Hello, my name is Dr. Roderick L. Roll, and today I will be talking about T lymphocytes. We are now on the third line of defense. Third line of defense is also under the acquired immunity. This is not immediate, this takes time to develop. Previously, I used a wrapper known as push and pee to describe how the macrophage or the antigen presenting cell will be activated. I first started talking about preparation. Preparation is to create the receptors, and the receptors were either MHC1, MHC2, MHC3, major histocompatibility complexes. Preparation is to make those receptors. I drew a little Pac-Man to illustrate this. You have MAC2 that's pink. That is a specific receptor that was created by this antigen presenting cell. Remember, it has a chromosome, it has a gene that is on that chromosome that codes for these receptors. And it is able to make different type of receptors. Next, using push and P, we talked about processing. So that antigen presenting cell will devour a substance that is foreign, and then it will process it. The phagolysosome will come into the cell carrying that particle, carrying that bacteria, carrying that virus, and then it would be degraded by lysosome. A lysosome fuses with the phagosome, creating a phagolysosome. The hydrolytic enzymes are released, and you partition, you fragment, you break apart the antigen. This illustration shows this purple bacteria, maybe it's a gram-positive bacteria that the APC is going to devour to process it. Push and P. We're on now presentation. Presentation is when the antigen presenting cell is now going to expose a piece of the antigen called an epitope on one of those receptors. It can be MAC1, it could be MAC2. This picture here shows you after the bacteria was processed, um, MAC2, the one that is pink, is exposing a particle, which is called an epitope. So it is presentation. So that was a summary of a previous slideshow we went over. And now we're going to specifically talk about the challenge. The challenge is when that antigen presenting cell binds to a T lymphocyte. The T cell or T lymphocyte is challenged by the antigen presenting cell when a synapsis occurs, a joining of the two. This is called an immunological synapsis. It is created, and that is when challenge starts. In the picture, I have a star beside the challenge. The MHC1 or 2 receptor of the antigen presenting cell will present the antigen or a piece of it called an epitope to a T lymphocyte. In the picture, I have outlined presentation that APC is presenting that fragment to the T lymphocyte. It will bind to one of two cluster of differentiation molecules contained on a T lymphocyte. So right where that synapsis occurs, there's a lot going on there. You have a cluster of differentiation. It's either CD4 or CD8. That is going to be the hand on the T lymphocyte. And the other hand that's on the APC is an MHC1 or MHC2. I tried to draw an illustration to show you what that would look like. The T lymphocyte has many hands. In the classroom, I use an example of a catcher's mitt, a glove. So the T lymphocyte has a T cell receptor or a glove that will bind specifically to dog dander. Also on the surface of the T lymphocyte, you have CD4 and CD8, cluster of differentiation 4 and number 8. The synapses, the joining of the two hands from the two separate cells, the, the APC cell and the T cell. So this immunological synapses is required to activate the cell-mediated response. Remember, under the acquired immunity, we have two major pathways, a T cell being activated or a B cell being activated. The B cell is a humoral response, whereas a T cell is a cell-mediated response. The cell-mediated response is only activated when the two hands join. Further, we're going to talk about the cell-mediated response as having different types of T cells, a T helper cell and a T cytotoxic cell. So from now on in the presentation, instead of saying T cytotoxic and T helper, I'm just going to say cytotoxic cell and helper cell. An antigen presenting cell containing the glove MHC1 will bind to the glove on the cytotoxic cell called CD8. To help remember this, the the when you take MHC1 times CD8, you get a value of 8. How does that look? The hand that is CD8 cluster differentiation 8 interacts with the hand of the APC called MHC1. Again, 1 times 8 equals 8. APC containing MHC2 will bind to CD4. Again, 2 times 4 equals 8. The CD4 is the hand with the glove that's on the helper cell. So in the picture, you have the T helper with the glove, with its creating the synapses with the APC MAC2. How does this look? The other hand of the T lymphocyte will bind to the MAC2. So the other hand is a CD4. So specifically, one type of T lymphocyte has that MAC2 and the other has the MAC1. So when that synapsis occurs, when the two hands meet, one from the APC and one from the T lymphocyte, that is referred to as challenge. We will now discuss different T cell subsets. If you look at the image, we have a T cytotoxic, we have a T memory, a T suppressor, or it can be called a regulatory, and we have the T helper. All of these types we will discuss, and they are created through clonal expansion, where you expand from one type of T lymphocytes to the different subtypes. We will now discuss only the ones involved in the cell-mediated immunity. They are going to be the T cytotoxic and the T helper. So out of the four subsets, only T cytotoxic and T helper will be involved in the cell-mediated response. Let's see how that is initiated. Remember, the T helper is one type and T cytotoxic. The helper has CD4, the cluster of differentiation 4, and the cytotoxic has CD8. CD8 binds to MHC1 on the antigen presenting cell, and CD4 binds to MHC2 on the antigen presenting cell. So how does this all work? So we're going to utilize this, we're going to use a scenario where someone gets herpes simplex 1. So endogenous antigens can activate cell-mediated immunity. And some examples of endogenous antigens are intracellular protozoans, bacteria, or cancer. This means those cells can go inside of your cell. Previously, we mentioned malaria and how a Plasmodium vivax or falciparum can get inside of a liver cell, hepatocyte, or can get inside of a erythrocyte or a red blood cell. This would be called an intracellular infection. We're going to specifically talk about a virus that has infected a cell. It's gotten into the cell and hijacked some of the enzymes that are required to make its proteins. So that is the scenario we're going to use to discuss this cell-mediated immunity. If it is an infected dendritic cell, it migrates to the closest lymph nodes. So in the lip, you have some cells that have been damaged by the or infected by the herpes simplex 1 virus. Dendritic cell devours some of those cells that are infected and takes them to the closest lymph node. While it's traveling towards the lymph node, a fictitious house where it needs to reside while it fights off this infection, it is going to process what it devoured. It processes that bacteria, that protozoan, that virus. In our case, the virus. It chops it up. It chops up the antigen, and it is going to present an epitope on MHC1 and on MHC2. Many of these antigen presenting cells have both MHC complexes. And immunological synapses occurs when the antigen presenting cell uh, carrying the epitope binds to a T cytotoxic uh, receptor. So in the picture, they've enlarged what we're talking about. So on that dendritic cell, on the right arm of the dendritic cell, on its glove, it is presenting a piece of that epitope, a piece of that herpes virus on MHC1. And you remember what binds to MHC1? CD8. CD8 is on the cytotoxic cell. So then it creates a immunological synapses, a joining of the two hands. This is called challenge. This is another illustration to show you what challenge looks like. A few cytotoxic cells are awakened but not activated yet. Simultaneously, on the left arm, a T helper CD4 is also activated when a microbial epitope is released on MAC2 receptor. So on the left hand, the glove, and the glove is a particle of the herpes simplex 1 virus, but it is now going to be exposed on an MAC2 receptor. And what binds the MAC2 receptor? The helper cell. So another immunological synapsis occurs on the left side of the dendritic cell. This is what that looks like. So that immunological synapsis is created. So we have just illustrated the challenge. The viral infected cell now releases interleukin 12, which stimulates T helper to differentiate into T helper 1. T helper 1 then secretes interleukin 2, which activates the inactive but awakened T cytotoxic cell. So in the illustration, you see dendritic cell releasing interleukin 12. It made T helper differentiate into T helper 1. Now T helper 1 is releasing a chemical messenger called interleukin 2, which is going to activate that cytotoxic to go through clonal expansion. Cytotoxic now begins to kill. It was awakened, now it has been activated. Activated cytotoxic cells release more interleukin 2, which self-stimulates proliferation of even greater numbers of T cytotoxic cells, meaning you had 5 cytotoxic cells that were awakened and then activated. Now that they are activated, some of them release interleukin 2, and interleukin 2, through positive feedback, makes even more cytotoxic cells be produced. And that followed the arrow number 4, self-stimulation. This increases proliferation, and it is called clonal expansion. During clonal expansion, a variation of cytotoxic cells will be formed. So we went from 5 to 5,000 cytotoxic cells, and let's say 5% of them are converted into memory cells. So now we have all these cytotoxic cells. So what? What do they do? They specifically kill. So they destroy in 2 ways. The first one we'll look at is perforin and granzyme pathway, and the next one is CD95 pathway. First, we have the perforins. They're going to make holes in the target cell membrane. Look at number 2. And those little red circles are called the peripherins. Those perforins are released from the cytotoxic cell, and they're going to be like these barrels that are dropped from an airplane, and they'll land on the target and puncture holes in the target. The target you see and with the red arrow I'm pointing to, the target is landing and busting holes in the membrane of that cell. Now, the blue circles, they're granzyme proteins. They're going to fall into the hole and then now reprogram that cell to commit suicide, a process called apoptosis. This picture B shows the same thing. T cell with perforin dropping those barrels. The barrels bombard the cell below it and puncture holes in the wall, in the cell membrane. The now granzymes are going to fall through the hole and deprogram and reprogram for that cell to commit suicide. And the bottom image, C, that is the CD95 pathway that also activates apoptosis. Other T cell subsets. We only talked about cytotoxic and helper because they were involved in the cell mediated immunity. Now we're going to talk about the others. We mentioned memory cells being made. So these memory cells can persist for months to even years. We know how those M cells were made. Now, what are they for? When a remembered epitope, MHC1 complex, matches the T cell receptor, the immune response is enhanced. So the memory cell remembers that original stimuli. And when it binds again, the immunological response is enhanced and immediate. Now that it has been stimulated again, it will expand from the memory cell into multiple cytotoxic cells that will then start dropping those barrels to destroy the cell. So T memory differentiates back into the TC clones and immediately recognize the epitope. This is quicker and it is enhanced, and it does not need the T helper cell to release interleukin 2. So you just eliminated a step, which makes it quicker. So that concludes the memory cell, one of the subsets. The next subset is the regulatory or suppressor. T regulatory cells make sure the killing signal is received in the correct sequence, or it suppresses the response. Sort of like the nuclear codes. You've seen those movies. You don't just want a crazy president to just push a button. So you want it to be some kind of chain of command. Somebody has to be requested to bring you a briefcase. Then it has another person has to put in the code to open the briefcase. Then once the briefcase is open, then there's a different person that has to be there to turn the key, and then finally the president to push the button. So you want to make sure the correct sequence is there before you start dropping those barrels, which are the perforins. So T repressor regulatory generally suppresses the immune response and promotes tolerance of certain antigens. The T regulatory prevents autoimmunity when self-tolerance is impaired. So the T cytotoxic on the left wants to start dropping those barrels on the thing that is not foreign to the far right. But that regulatory is there to slap it, to say, no, you are not allowed to release those barrels to destroy that cell. It is not foreign. So the regulatory is needed to suppress that response. It makes sure the immune system does not inappropriately target the normal biota. That concludes the regulatory. This is just an overview of what we've covered so far to show you the intricate uh, hybridization or synapses or immunological synapses that occurs between these two entities. They're very involved in the immune response.